hello everyone. Thank you for being here after your lunch. And so this first session of the afternoon, I'm going to talk to you about higher organisms. So here you see the development of a chicken embryo, the first 2 days that it is growing. So you see the formation of the head and the elongation of the tail from the posterior. And this is the topic of our discussion today is going to be mainly focused on how the tail is growing in vertebrate embryos. So let's first look at different mechanisms that a tissue can grow. So there are many mechanisms that tissues use depending on what works better for them. And most of the mechanisms that you can find in literature and have been studied are focused on epithelial tissues. For example, here you have the elongation of the root of the plants, which in this area that is pictured here, the mechanism that works is that you have the tissue that only at one side the cells are dividing. So you have localized proliferation that makes the tissue grow. Another way that you can imagine a tissue can grow is just if it is stretched and the cells change their shape, which is basically what C. elegans is using for growing. Here you see the membranes of the cells are marked, and you see that are really stretched. So this is the cell elongation mechanisms. And another one that most of you probably have heard is the convergence extension mechanism that, for example, we see in the germ band expansion in Drosophila, where once the tissue upon an external force application, the cells in a tissue start to change their neighbors, and you have elongation in the direction of the applied force. Another mechanism that you can imagine that I haven't put here is that there is an external force that is applied to the tissue, and this favorizes the division of the cells in that direction. So you have like a directed cell proliferation, which also can give you tissue expansion. Now, if we look at our chicken embryo, here you see all the stages that go from the first stage to the end of the 21st day. You see that there is a big change in the shape of the tissue. The first few hours, like let's say the first 1 day, the embryo goes from a ball of cells that has no shape, and little by little you see the appearance of the anterior-posterior axis that you see here. And these steps also, most of this elongation of the tissue can be explained by the convergent extension mechanism. But then we reach to the elongation step, where you have the embryo, the axis is already formed, and the tail is growing. The movie that you saw in the beginning, the end of the movie, is basically these steps. And these steps are, we cannot explain them with this convergent extension mechanism, because as you will see, it doesn't follow the neighbor exchange mechanism that I talked to you about. So we will try to a little bit look more carefully on what happens in these stages. So why are we interested in studying the axis elongation in vertebrates? This is the embryo of a human at 22 days. So it's quite small, it's like 3 millimeters. And during the elongation stage of the embryo, another process happens that is called somitogenesis. Basically, the formation of these packs of cells that you see them quite separated in a very regular manner from each other. So these somites are the precursors of what we will later become your skeleton, your bones, your skeleton, and your muscles, as here I have shown you. So anything that goes wrong in the formation of the somites can have an impact on the formation of the skeleton. So one of the diseases that some of you might have heard of is the congenital scoliosis, that is a malformation of the spine. So this is something that earlier in the development of the embryo, you have a problem in elongation, in somite formation, and later this can give rise to vertebrae that are not well packed on each other, or you have some other malformations. So this is why we want to study this mechanism. So, of course, we cannot use human embryos for our studies, so we use other model systems. So one of the models that has been quite used is the mouse. Of course, mouse embryos are quite common in laboratories, but then you can also use chicken embryo. That here there is a very recent review came out on this topic, and chicken embryo model system, and why this is very useful in the labs, and it has quite been in the market for a long time. It's not a new, new discovery. So the advantages that you have is that, first of all, the embryo develops outside the mother. So if you want to study the embryo development, you don't need to sacrifice the mother, which is basically what happens if you use, for example, mouse model. It's quite accessible, so you can manipulate the embryo and let them grow in their environment and look the next day and so on. I will show you how. And this is one big advantage. Economically, it's very interesting because it's quite cheap and you don't need animal facilities in your lab. You can get these eggs from the farm, and it's quite easy to have them. And then you can also imagine to have a lot of embryos that are well synchronized, because you can incubate them in a certain time, and then you can kind of take them on the stages that you need. So quite practical model system. And so how do we do our experiments? There are 3 techniques for visualizing embryos. Either you just open a window on the eggshell and you leave the embryo develop on the yolk. Here you see already the veins, blood vessels have formed, and you can see even the heart beating and so on. So, and then you can do your manipulations, for example, if you want to inject some drugs and so on, and then look at them under the microscope. If you need to leave them grow, what you do is that you close the eggshell, you put it back in the incubator, and they will grow, and then you can look later on. So this is one technique. The other one is you take it out. It's basically the same thing, but probably this gives you better visualization for microscopy. And the third technique that has been around for a few tens of years, I think 10, 15 years, is what is called the new culture. And this is what we use in our lab. Basically, in this stage or earlier, you can put a filter paper here, like a filter paper that you use in your chemistry labs. You make a hole in the middle, stick it on the embryo, wash it in PBS, and you put it on a glucose gel. And with this glucose and agar gel, they can feed themselves, and then they can grow for up to 2 to 3 days. So you can look at the development of the chicken at these stages. So again, it's very practical for microscopy. This way you can do very nice microscopy. You can even track cells if you inject some constructs that mark the cell nucleus. You can do cell tracking. You can do micromanipulation. The only limitation is that this is good for young embryos. The embryos, when they grow, they cannot fit in this filter paper, and then you cannot do your experiment on them. Okay? And so please interrupt me if you have some question or some comments, okay? All right, so this is the technique that we use for our imaging. And this is, so I said that is the comparison between the human embryo and chicken embryo. So you see they quite resemble to each other at these stages, even though it's not the same day, so they grow faster. Another advantage that chicken has for our studies is that, as you see, the tail region, which is called the pre-somitic mesoderm. So this is a mesenchymal tissue that gives rise to the somites. So it's quite large. So for manipulation, it's very practical. Here you see that for the human embryo, it's quite small, and the mouse is also quite small. It's PSM. So you can have a less material to work with. And the chicken embryo, another advantage that it has is that it's two-dimensional. It's quite flat. It's not two-dimensional, but it's quite flat, and it's growing quite flat. So for imaging, you don't have a problem of the embryo coming out of your field of view and trying to do 3D imaging to position cells, etc. Okay, so now that we have our system, how do we do our experiment? So here you see the growing of the embryo for 2 days. You see the formation of the somites and the elongation of the tail. To characterize this elongation, what we do is that we take one of the somites as our fixed point, so that we can track the growth of the posterior part. And we take the end of the neural tube, so I will come back to this later, so this black line in the middle, and we see how this elongates over time, and you see that it's almost linear. So for a very long time, the embryo grows at a constant rate. And as we go further, as the embryo grows further, then we see that it curves and then it stops. The elongation stops, and we don't have any more growth in the vertical direction. Okay? So this way we characterize our elongation. And then what we want, so here, first before going to data, what I want to tell you is what are all the tissues that we have in the posterior region, because I talked to you about presomitic mesoderm, which is this mesenchymal tissue here shown in orange, but you have other tissues around. So in the middle, we have the neural tube that later will give rise to all the neural system. The somites are at the front, and if we look at the cross section, we have the ectoderm and the endoderm. So you have this kind of a package where in the middle you have the PSM, and then you have all the other tissues that are all attached to each other by extracellular matrix. Okay? And so if we look at how these different tissues grow over time, this is a work done by Bertrand Benazera, who was in our team. And what he saw is that the PSM over time grows much more than all the other tissues. So he just marked over in different stages, he marked the borders of different tissues, and he kind of compared them to each other. So the PSM is a quite important tissue in this process of elongation. And if we look a little bit more carefully what happens, so what we know that happens during this elongation process is that you have this mesenchymal tissue at the posterior. These cells, as they go towards the anterior, they start to adhere to each other more and more, and they form an epithelial tissue. And this epithelial tissue later detaches and it forms the somites. Okay? And the way that the tissue grows is that it adds new cells from the posterior part. So new cells are added here. They create a gradient of growth factor, FGF, and this controls the motility of the cells inside the PSM. Okay? And so we have this mesenchymal to epithelial transition all along the PSM, so from posterior to anterior, tissue compaction at the anterior, and the addition of the new cells. And this continues for a while until the elongation stops. We don't know what makes the addition of the new cells at some point stop. So this is what limits the elongation at the end. Okay? And then, so one question that people want to know is that, okay, so if we have the new cells that come from the posterior, do they kind of migrate towards the anterior and end up in somite? So in this case, the cells would end in the somite in an orderly manner. So what they did is that they marked different regions of the PSM with a dye eye. So this is a dye that marks the cell membrane. So here you have different regions. They marked them with different colors, and they looked at how they ended up in the somites. And they saw that there is no correlation. It's not that the cells that arrive first, they end up on higher somites. So at some point, these cells mix up with each other, and then it's more or less random the way that they end up in the somites. Okay? And so Bertrand, another experiment that he did was that, okay, he wanted to know which part of the embryo, the posterior part of the embryo, acts more in the elongation process. So for this, he just did with conceptually an easy experiment, which was he put a gel in different, so he did a laser ablation, and he put a gel in the incision that he made, and he looked how the embryo grows without having that tissue present. So if he was ablating the axial posterior part, so the tail bud region, he saw that the embryo doesn't feel it that much, so it still continues elongating almost at the normal speed. But when he was ablating the PSM of the paraxial part, so the two sides of the embryo, what he saw is that the embryo stops growing. So can I play that? So the embryo almost stops growing. So the conclusion from this experiment was that these parts of the embryo, the tissue is the most important in the elongation process. So we had to study a bit more what happens in this region. So this is just to show that he also looked in different regions, and as you can see, is the posterior PSM that drastically reduces the elongation rate of the embryo. So then, of course, we wanted to look more at the cellular level, what happens. So we use a technique that is called electroporation. This way, you inject a fluorescent DNA at an earlier stage to your embryo, and with this technique, you mark one population of the cells because you have like a salt and pepper labeling of the cells. And you can see here that you have, for example, here it's around 40% of your cells that are marked. So then you can track them and see their behavior in different conditions. So what we get is that so we track our cells, and we divide for analysis purposes. We divide the posterior PSM to 3 regions. So we will call it anterior, mid, and posterior, and we calculate the mean square displacement of these tracks. So as you can see, there is a big difference between the posterior cells and the anterior cells. So there is a graded behavior in their motion. And to analyze this MSD, we considered an MSD of this form, which has 2 parts. So we have a diffusive part for the cells, and we have a drift term for the tissue. So the tissue has a drift, so it's growing, but at the meantime, the cells inside this tissue is diffusing. So with this fit, we got the data for D and V. And what we see here is that there is a difference between the diffusivity of the cells at the anterior of the embryo versus the posterior. And the same for the velocity of the tissue. Okay? But now what happens if I act on the activity of the cells? So the only way that we had at the time to do was to use either drugs. So we use blebistatin to affect the cell motility. And we saw that this diffusion gradient of diffusion is changed when it is treated by blebistatin. Yes?
Is there any motivation behind why the diffusive coefficient should be different in different regions of the...
So we can talk about it. Why? So there is no actual reason why it should be. This is what we see. But you can imagine it could be because the cell density goes higher at the anterior. So for cells to move, it is more difficult. So this will be kind of a viscosity problem. It can be because cells are adhering more to each other. So there are like things like this. Okay? Any other questions? Okay, so this is the drug treatment. And if we reduce the temperature, we also see that there is a difference in the diffusivity of our cells. These experiments, I should tell you that in research, sometimes it happens that you do experiments by accident. And one day, what happened was that our heating system in the lab was broken. We had all prepared the experiment to run, but then suddenly the heating system broke. And then we put the embryo to just see what happens in room temperature. And by surprise, we saw that we still have cell jiggling. So diffusivity was still there, but the embryo was not growing in length. So we said, OK, so we will do more systematic study of varying the temperature and see how is this correlated with the elongation rate of the embryo. And so this was what we found. And then, so now we wanted to see how is this correlated with the elongation rate of the embryo. So very simply, what I defined was what I call the macroscopic gradient of diffusion. Basically, it's the slope of this line. And you see that it changes with the different conditions that we had, as you saw by the lines. And now we correlate this with the elongation rate that we measure. And what we found is that everything collapses on the same line. And here what is interesting in this data is that, for example, here you have two different treatments. You have treatment with blebistatin, which is the drug that acts on cell motility, and you have a reduce of the temperature, which is a completely different mechanism for acting on cell motility. And in spite of that, you get the same elongation rate for your embryo. So this is one thing that this data is showing. Another one that it is showing is that you need to have a special, your diffusion gradient has to be larger than a specific value so that your embryo can grow. This is why that at room temperature we have gradient of diffusivity, but it's very small, so the embryo is not growing.
Sorry.
Yes?
Why do some of the points have error bars on both the axes, but the other ones don't?
So for here, probably I haven't had a lot of variation in the velocity that I measure, because these are all measured values, you know. Yeah? Okay, after these experiments, we wanted to go a bit more in detail of the profile of this diffusion and velocity gradient. So instead of dividing our PSM in 3 parts, we did smaller groups of cells, so smaller windows. And here you see that it's not that linear. It's a little bit more like exponential decay for these 2 parameters. So we wanted to see how, by having this kind of data, we could explain what is happening for the elongation of the embryo. So we turned into, and then another thing, what I wanted to also mention, that we talk about this diffusion. So we can define this diffusion by what you already know, which is the ratio of the energy that you put in your system and the dissipation that exists there. So here we should just note that we put an effective temperature. So it's not real temperature. This is always, sometimes this comes up, that if you change the temperature, so you're changing the thermal energy, but that's not the case. It's an activity of the cell that you are changing. And the dissipation comes from what I talked a little bit about, like the viscosity that can exist inside the tissue.
So what's the role of the growth factor group?
So the growth factor controls cell motility. That is what creates different motility for the cells at the posterior versus the anterior, because you have this gradient here. So if you have a high FGF concentration, your cells are more active, they move more, and then if you reduce the FGF, they move less.
So this is like an additional factor to the viscosity effects?
Yeah. So you can, it's actually, since we don't know exactly what is happening, we can either consider that it's the cells in FGF that are moving more versus less concentration, or you can have a passive viscosity contribution, or both. All right, so then with this data, we went to see our physicist friends, theoreticians, and they put together a simple model, like a first, let's say the alpha version of the model, to explain what happens. And the considerations of this model are the following. So I'm going to tell you just what we put in the model, and then let's see what happens. So they consider that we can define a density rho for the active cells. So this is not the density of all the cells, because the density of the cells are, of course, you can have active cells and immobile cells in a region, okay? And it's like a switch that with a probability, a cell can be active and then it can suddenly become inactive. All the active cells, you give them a diffusion that is a single cell diffusion. They consider that for the moment the PSM has a uniform viscosity, and it's yes.
You said the cells start to make contact between them when they go to the anterior.
Yeah.
So it should increase the viscosity, no?
Yeah, we will go there. For this part, they didn't put that into the model, but probably in later versions we have to take that into consideration.
becomes inactive? Just the fact that it becomes like...
So basically what we think is that depending on the FGF concentration, there is a probability for cell to be motile, like amoeboid motile, or suddenly become immotile. You know?
So it's not a gradient?
It's not a gradient in their motility. It's not that slowly they slow down, they slow down. They are moving with certain speed and then they stop.
Because you have the kind of gradient of diffusivity, so...
Yeah, so what I measured, and the way we explain this, is what I measure is the movement of all the cells, so mobile and immobile, and I measure something that is the mean, which is basically what I have put here. So the measured diffusivity is the single cell diffusivity and the ratio of motile cells to the total cells. Right? And then, of course, there is the injection of the new cells that you have at the posterior. So they did the two models for us. One model that I will explain in the next slide for you, but they also did a cell-based model just to show that with these same considerations, you can have an elongation of the tissue at the towards the posterior direction.
Just again, you just have two states. Either you're mobile or not.
Yeah.
And when you're mobile, you're diffusing at the same speed.
Exactly. Yeah. Yeah.
Is it just for the model, or you see it?
So it's a tricky question. Yeah, it's a tricky question because in vitro, we saw that if we would take PSM explants, dissociate the cells, and plate them on a surface, depending on the region that we take them, we can have a different ratio of motile cells versus immotile cells. Okay, so this is something that gave us this hint that maybe there is something that changes the switch between motile and immotile depending on the position on the axis. Okay, but in vivo it's very difficult because because these cells are in contact with each other anyway. So even if one cell changes state and it's not actively moving, it's pushed by the other cells. So in the end, you measure something that is the mean of all these movements.
If you track cell in vivo, you have a bimodality?
No, but for this reason, because everybody moves, you don't get a bimodal distribution. It's not that some stay immobile and the tissue moves together. So you don't know the one that is not actively pulling, it's just pushed by other ones. Right. Okay, so the continuum model is defined by this equation. So the mass balance equation that is given by these different terms. So the change in the density of the motile cells, the diffusivity, the advection, which is the same as the one that I put in my analysis, and the decay, which basically says that it exponentially it decays as you go towards the anterior. And the force balance is a simple, by taking a simple equation, like an ideal gas model for the pressure. So you apply a pressure to the tissue that has two dissipative terms, the viscosity of the tissue inside, and you have the friction with the neighboring tissues, which are here as we model the one side of the PSM is the neural tube at one side, lateral plate at the other side, and the somite at the top. Okay? So then with these two equations and this activity, so the pressure which is proportional to the activity of the cells, this activity is what we talked about, is this FGF concentration. So this is just the way we explain it in the model. And they fit our data with their model, and by taking, because for this part we just did some preliminary experiments to get the viscosity of the tissue. So by putting the viscosity of 10 to the 4 Pascal seconds, they could get these two terms, so alpha rho 0, which is around 1 Pascal, and the resistance of the wall at the posterior. So as the embryo is growing, there is a resistance from the tissue in front of it, which is measured here. So now we are developing some, I'm not going to go through it, but we are developing experimental techniques to look at, measure these values experimentally and see if these predicted values are correct or not. Okay. Okay, so that was for the active part, which we tried to model. But then, as many of you already asked, what happens to these rheological properties of the tissue? Because we ad hoc we put a viscosity constant, but maybe it's not the case. And indeed, for another system, which is here the zebrafish embryo, and it's a paper that came out recently, a beautiful work at Campus Lab. What they did is that they put oil drops inside the tissue. These are ferrofluid drops, and they can deform them by magnetic field. And you see here, they deform the oil drop, and then they release it, and they see how it retracts. And from this, the movement of this, analyzing the shape of this droplet, they could see that there is a yield stress gradient between the anterior and the posterior of the embryo. Then they used a lot of modeling, and for explaining the elongation of the embryo, they were obliged to put a gradient of the viscosity so that the elongation takes place. I'm not going to go through their work, but if you are interested, please look at this paper. So for our experiments, we had other choices for measuring the rheology of the tissue. Here I'm just going to talk to you about few techniques that exist in the field that people have used. The classical one is the parallel plate compressor that you can see. It's so this is the first papers that came out that they actually measured values to measure the surface tension of the cell aggregates. So with this technique, you can use basically the surface tension. You can sometimes, if you analyze well your data, you can also get the viscosity. Then you can have indentation techniques, which is basically giving you the elasticity of your tissue. This can be like an AFM. You can just deform your tissue, or you can send a jet, like hydrodynamic jet, to. So this paper is actually that deforms the tissue, and from the deformation you get the elasticity. We have the laser ablation technique that is mostly used for two-dimensional tissues. It gives you the anisotropy in the stresses, because from the way that the tissue relaxes, you can see which direction there was a higher stress than the other. This technique, it's quite cool. What they did is that they stick a bead, a magnetic bead to one side of the tissue, and with a big magnet, so it's very artisanal, as you can see. You can come with this magnet, and you can pull on this tissue. By the deformation of the tissue, you measure its elasticity. So this is this one here, you have a gel drop inside an aggregate. It's similar to the oil droplet that I showed you. Again, you can measure the stresses and so on. And what I'm going to talk to you about is the pipette aspiration, where like similar experiments you have probably seen on single cells or on GUVs, where you aspirate your material inside in the pipette, and from the deformation of the material, you can get its rheological properties. So how it works, it's shown here. So this is on model tissues. These are cell aggregates. So you prepare a solution of cells, you let them stick to each other, and after a while you have a nice ball of cells. And so you do your experiment. What you measure is basically the advancement of the tongue inside the pipette. So the applied force is given by this. So RP is the radius of your pipette, delta P is what you have applied, and delta PC is the critical pressure. This critical pressure is related to the surface tension of your aggregate, because the fact that your aggregate, when you make it, it takes a spherical form, it shows that there is a surface tension that tries to minimize its surface and takes the spherical shape. So it's given by this equation. Gamma is your surface tension, and so on. So the rest is here. And so what we got for our experiments was that you have a curve looking like this for the aspiration regime at a constant pressure. And once we release the pressure, where the pressure is put to 0, you have the retraction of your tongue. And both of them, they showed you an elastic regime that is given by this rise of your curve, and then you have a constant flowing regime that gives you the viscosity of the tissue. And any questions at this point? No. Okay, so just concentrating on the viscous part, your viscosity will have two terms, because you have this aggregate that is entering the pipette. There is kind of like a plug at this port side that you can see here, like the deformation of your tissue, given by this equation. Eta is the viscosity, and L dot is the speed of your advancement inside the pipette. And the second term is the friction with the wall. And we consider that this term is negligible because we treat our pipettes with PEG, which kind of prevents cells from sticking to the pipette. If you don't do this treatment, your curves don't look linear anymore. They start to curve. That shows the slowing down of the advancement of the tissue. And so this gives you the relationship for your viscosity with respect to your applied pressure. And so you have two terms, unknown terms. Let's go back. Yeah, here. So for example, for knowing your speed of advancement, you have two unknowns. But since you have an aspiration curve and the retraction curve from these two equations, you can get your viscosity and the surface tension. So the viscosity as a function of the applied force, this is the stress, you see that there is no relationship. It's not, you don't change the viscosity. So it means that there is no fluidification of your tissue, like so the viscosity stays constant, sorry. And just to compare, you see that your viscosity, we talk about tissue viscosity, it's a very large number because already honey is a 200 Pascal over Pascal times second, which is quite viscous material already. So this was quite encouraging, and our result here was very close to what people have used with other techniques. So it showed that even though you are applying a pressure, you don't, that doesn't affect your measured value. But surprisingly, what we found is that the surface tension that we measured depended on the force that we were applying. So if I had an inert material, that surface tension should be a constant, and it shouldn't vary with the force that we apply. But in these tissues, we saw this. And we explained it by the fact that upon application of a force, your cells react. So they reinforce their cell-cell contacts or their cytoskeleton reorganization. And this increases the surface tension. Because the surface tension is related to two values. So it's the cell-cell adhesion and the cortical tension. Okay? And this is also a proof of this behavior, which is here we apply steps of pressure, and we look at the dynamic of the aspiration. So 1 kilopascal, 2 kilopascal, up to 5, and then we go down. And what we see is that when we come back to, for example, 3 kilopascals, our tissue is no longer flowing, which means that here it has changed its surface tension. It has stiffened. So by knowing all this, we go now from aggregates to embryos. So how can we apply this technique to measure embryonic mechanical properties? So this work was done by Arthur Michaud, who was at the time a PhD student. So what we do, the way we approach this, is that we dissect our tissue outside of the embryo, and then we divide it to 3 regions, the posterior, mid, and anterior, and we do our experiments on them. So we can do different type of experiments. So here I show you 2 very classical experiments on tissues to show that they behave like liquid, like they have a liquid behavior. You have a fusion. If you put 2 explants next to each other, after some time they kind of merge and they make a sphere. And if you take a tissue that is elongated at the beginning, after a while it takes a spherical form. So we can analyze a little bit better their dynamic and get some information about their mechanical properties. Yeah, so this is a picture that for the moment we have if you have cells that have two different adhesivity to each other. So the adhesion molecules that are different from each other. If there is a perfect adhesivity, it will be what we see, which is basically at the end, you have a nice spherical aggregate. If one of your tissues is much more adhesive in between each other, so you have higher number of adhesion molecules from one tissue than the other, then the most adhesive one will end up at the center, and you have a layer of the other tissue around it. Sometimes your cells don't adhere to each other. In this case, you have what we call a weak adhesion. So your tissue at the end will stay like this, or won't adhere at all to each other. So this already gives you an indication of what is the adhesivity of the cells to each other. Okay, so to analyze our data, we take explants from the posterior and the anterior. And here, let's see if this movie will show. I don't have my mouse. Yeah, so you see that for the anterior region, there is a lot going on before the explants merge to each other and make a sphere. So there is the dynamic is not at all the same. So let's go. So to analyze this, what we do is that we measure the radius of the neck over time, and we use this equation to get this value, which is gamma over eta, surface tension over viscosity, for these fusion experiments. And what we see is that there is a big difference for the anterior explants versus posterior explants. So the gamma over eta is very different in these two regions. Interestingly, if we take an explant from the anterior and put it next to the posterior, we don't see any fusion anymore. So the final form resembles to what they call a weak adhesion scenario. So the way we explain this is that at the anterior of the PSM, your cells are starting to express specific cadherins that doesn't exist at the posterior. So these two cadherins, they don't find each other, and the cells don't merge anymore to each other. Okay, so another experiment is the rounding experiment. The same way, we take our elongated tissue, we look at how this tissue aspect ratio changes over time, and you see a big difference between the anterior explants and the posterior explants. So again, we use an analysis. So here we did the experiment with different radii, because so this is the equation that explains the dynamic of the rounding. And you see that it depends on the final radius of your explant. So the larger tissue that you take, it's going to take longer to round up. And again, so we have this term gamma over eta for our explants that we can find from the slopes of these lines. So you see a big difference between the anterior and the posterior. So blue is the anterior, sorry. So it's much slower to round up than the posterior part. And we get a very similar behavior as the fusion experiments. So we have the gamma over eta for posteriors is 3 times larger than for the anterior. So this can say either the surface tension is higher at the anterior, which is a bit weird, or the viscosity is larger at the anterior, which goes more towards what we were thinking originally. But well, this experiment only gives us the ratio of these values. So now, how can we get these values separately? So I don't know how I am doing for the time. I'm 5 minutes, yeah. Okay, so we came back to our pipette experiment. So First of all, we were very happy that it already worked, because this is, as I said, this is a mesenchymal tissue, and mesenchymal tissues are very, very fragile. They are not comparable to the cell aggregates that I showed you in the beginning. So the curves look similar, so we can analyze them and get the values for our viscosity. And we can also measure the surface tension separately. So the way we do is that you take your aggregate and you start to aspirate in very small pressures. So you see here, for example, your deformation is below the radius of your pipette. So you increase your pressure, so on and so forth, until you get to a deformation that is the same size as your pipette radius. At this point, the pressure that you have applied is the critical pressure of the aspiration. So directly from the Laplace law, you can get the surface tension. So see, this is what we call the direct measurement of the surface tension. So we didn't see any gradient in the surface tension for the PSM. For the moment, I say, because as you see, we don't have that much the data neither, but we didn't see any tendency. Then when we do our aspirations at different positions, so for one embryo, so you take one embryo and you do your aspirations in different regions, we saw that there is a clear difference in the aspiration dynamics. And it gives a factor 3 between the viscosity of the posterior and the anterior. So this is already quite encouraging. Except that you are again working with in vivo systems, very complicated. So when we did more experiments, then the tendency was much more fragile. So you see that here, if we put all our data together, the gradient is very weak. But if you take embryo by embryo, so if individual embryos to compare to each other, we see a slight gradient for the viscosity from anterior to posterior. So this is a work in progress. We are trying to understand, because there is a slight discrepancy between the rounding results and the pipette results, and this we have also seen with other tissues. So it's something that we have to figure out if it comes from the experiment or it's a modeling issue, a factor 2 somewhere is missing or something. So this we have to work on it. And with this, I just sum up my talk. So I talked to you about the role of cell motility in elongation, the way we analyzed it. So we are doing some gain and loss of function experiments to see how this will affect the elongation. I presented you a little bit of the rheology of these tissues, and we are now working on measuring the elongation force by a cantilever setup. And what I put here, the new directions, is basically what I'm trying to develop here at Curie, because this work was done when I was in other places. And so now I'm trying to work on the mechanics of segmentation and somite formation on the same system. And I just take 2 minutes to tell you why I want to work on this. So this is an electronic microscopy picture of the somites. And I always find it fascinating, the way that your PSM here suddenly starts to form these balls of cells, and later they separate from the PSM. And if we look at the cross-section of the somite, you see the cells that are very nicely aligned at a layer, and then you have other cells inside. So how mechanically this is possible, that the cells here that are all in all directions and so on, little by little, they get a structure and they separate from the PSM. This is what I would like to understand. And the indications that we have is, first of all, for example, the reorganization of the actin during this somite formation. Here the actin is labeled, and you see that you have a high accumulation of the actin at the inner layer. So there is a tension built up around the somites, and this excess tension could be the reason why these packs of cells separate from the PSM. So, okay, so this is just maybe in 5 years I can tell you more about it. And I would like to thank you for your attention. It was not easy after the lunch, I know. And so my team, where I am right now, in Sykes plus Tino team, and all the people that worked with me on this project. And thank you very much.